Well, you have done several online adaptive lessons over the cell membrane and the importance of the cell membrane. And if you've done the cell membrane adaptive lesson, you've already seen these types of transport. But in case you haven't, we wanted to review a little bit and get on paper some of the types of transport that are allowed by the cell membrane. So I'm going to start actually with your notes backwards. We're going to do this page that has these little pictures at the bottom first, and then we'll go back and do the page that looks like this second. So we're going to do this one first. So obviously the main structure in the cell that regulates transport is the cell membrane. And you've already talked about the fact that the cell membrane is made up of phospholipids and proteins. And then every once in a while you'll have those chaperones, those cholesterol molecules, and several other markers on the outside of the membrane that help the cell for cell-to-cell recognition. So you kind of already talked about those. We've already talked about the fact that water has to go through water has to go through those proteins. I noticed in the adaptive lesson that they actually had it going through the phospholipids, so apparently they haven't got the latest and greatest information on that, but water has to go through those through the proteins, as well as large molecules like sugar. And then ions, things that are charged like sodium or potassium. And then things that can actually squeeze in through the phospholipids we've talked about in and out are those gases like carbon dioxide and oxygen. So we've already reviewed all that a little bit. And so as these things go in and out of the cell, these specific ones that we're looking at right here are all types of passive transport. Well, except for ions. We'll put a star by it. Passive transport, when you think of things that are passive, you think they don't really require, they just sit back, they kind of sit back and watch things happen, not too worried about what's going on, they're like chilling, right? And that's the way the cell is. The cell does not require any energy to bring these things into the cell, like the oxygen, the water. They just naturally move. Now in passive transport, these molecules are going to move, and here's your big thing, from high to low. So like if you were to take a balloon and you were to pinch off the balloon, and you have a lot of air in here and it's really, really tight. If you were to let go of that balloon, you would say that this is a very, very high pressure area, and that air, if you let go, would move to an area of low pressure. And so molecules tend to do that as well. And there's three different kind of categories in biology of these molecules as they move from high to low. The first one is diffusion. Now diffusion is any time molecules move, just generally any kind of molecules, not water, because it has its own little term right here, which is osmosis, but the movement of any molecules. So you could say maybe somebody wearing perfume, really strong perfume. You could smell their perfume across the room. Maybe a candle. Maybe if somebody farts or passes gas and then they walk by you, don't you love that? Perhaps even like in your lab, in the starch in a baggie lab. What was able to move across that baggie without any energy? Like we didn't electrocute anything, it just kind of moved. It was the iodine. And the iodine just kind of moved into the baggie. So when molecules move from an area of very, very high to low concentration, any kind of general molecules, that's called diffusion. Now when molecules specifically move across of water, when the water molecules move across the membrane, or even a Ziploc baggie or anything like that, that's called osmosis. Now you've probably seen that movie Osmosis Jones. Well, Osmosis Jones, or maybe you've heard the expression, I'm just going to learn my biology through osmosis. Both of those terms are actually used completely incorrectly. Osmosis Jones is not the movement of water. He's actually a white blood cell. And I'm just going to sleep on my book and learn through osmosis would imply that your book is made of water and it's actually going to just diffuse into your brain. And so that's not really true either. So anytime water moves, that's called osmosis. So you've probably heard of people being stuck out in the ocean for days and days and days. And when they're stuck out in the ocean, they're told not to drink the water. If they're in the water, they're in bigger trouble than if they're in a raft. But ocean water if you are stuck out in the ocean, you will actually, whether in water or you decide to drink the water, you will actually dehydrate because all of the water will leave your body, not because you said, hey, water, leave, but because it's trying to maintain a balance with the outside environment. What's that called? Homeostasis. So ocean water actually takes the water out of your cells. Another thing that takes water out of your cells is alcohol. If you drink alcohol, which you should not because you are not old enough to be drinking alcohol, or colas are very, very, very high in solute concentration or not water, then the water in your cells will leave out of your cells as well to try to maintain the balance of these high solutes in your blood vessels. So water is going to move depending upon the outside environment of your cells, and when it does, that's called osmosis. And we talked about, just remember, when water moves through, these proteins up here are called aquaporins. Now, the third type of passive transport is facilitated diffusion. Now, I don't know if you've ever heard someone called a facilitator, but for a while, teachers, we were trained to be facilitators. And they told us that we weren't supposed to be the ones actually doing all the work, that we were supposed to be helping you learn, helping you do the work. And so for a while, all of us teachers were actually called facilitators. And we still should be today, but it was very trendy for quite a while. So if you look over here at the membrane, what is facilitating or helping the movement of these molecules? Well, these proteins right here, these doorways. And so those proteins are called integral proteins because they go into the membrane. Now you'll notice these molecules, be it oxygen or carbon dioxide or whatever they happen to be, if you count them up, there's 1, 2, 3, 4, 5, 6, 7 on this side of the membrane, and 1, 2, 3, 4 on this side. So where is the concentration the greatest? Good, it's on the outside. So if it's high over here, molecules are going to naturally move from high to low. And we said that's called diffusion, but in the event that they need to go through a helper or a doorway, it gets a little bit more specific, and it's called facilitated diffusion. So things that actually have to go through a doorway would be like glucose, your sugar. Your sugar is pretty big. Amino acids. What are amino acids going to go through the doorway to come out and make? Well, they're eventually going to make proteins. And so these are bigger structures. They're not ginormous, but they're bigger structures. Now, we're going to go back to that other page that I told you about that we were going to do second instead of first. And so when molecules tend to move as a result of diffusion, one of the things that you got to realize is that they're going to move. Molecules don't stay still. And so I like this little picture of these little guys, which you can't see them right now, them moving. And they just keep moving, moving, moving. And any kind of molecules, people are going to move, organisms are going to move, things are going to move. But I wanted to know one time, why do molecules move? Why does perfume go through the air? What actually causes it to do so? And I found this real simple animation that kind of explained it. And so you could see right here in the middle, let's say this is perfume right here, just that you just sprayed maybe on your shirt. And this is the air around it. And so as you spray perfume on your shirt or maybe into the air or potpourri or something like that, look at what happens to the other molecules around that perfume. What happened to them? Well, the other molecules in the air are moving constantly, and when they bounce into the molecules you just sprayed or that were on your shirt, it spreads out those molecules because molecules are always moving. And I guess when they're frozen, they slow down. They don't move as quickly, of course, but they're generally moving. And so I like that animation because it helped me to understand why do they really move. Now, in order for there to be a move, not really, but one of the things that you'll see in diffusion that powers movement, we said, was a high concentration in one area and a low concentration in another. And I kept saying concentration, concentration. Well, concentration isn't like to think deeply. It is how much of something you have. So maybe the quantity. Like, for example, in PowerPoint, if I wanted to change my background here in PowerPoint, well, I'd actually have to hit the right thing, and I wanted to make it a different color, and I wasn't really sure which color I wanted. I could go over here to colors, and I'm like, I don't know. Well, you'll see here that there is a higher concentration of black down here than up here, or even blue in this case. This is called a gradient, which means it changes levels. And so when you hear the word concentration gradient, you should think of those color changes. And so we could do like a little drawing right here if you want. And you could take your highlighter, and just get it lighter and lighter and lighter. So you have a gradient, or a high area of color here to a low here. Now, when we're looking at water, so this page right here, this whole page right here, really is talking about not always the movement of water, but we're usually talking about what happens to water as a result of there being a concentration gradient. Not always. Water is not the only one that moves, so don't get that misconception in your brain. So when you look at these three diagrams, for example, let's assume that these little dots right here are salt, little pieces of salt. And you'll notice that there is more salt inside the cell. So this is inside the cell, each of these diagrams, and this is outside the cell. And you'll see in this diagram there's more salt in, so more here, less here. And what about in this one? What do you notice about the little dots? Well, they're almost equal. They're almost the same. And what do you notice about this one? Wow, look at all the salt outside here. There's a ton of salt. So there's a lot more on the outside than on the inside. Now, the salts can move, but the sodium specifically, usually has a charge associated with it, and it's a bigger structure, and there's a lot more involved. So more likely than not, it's not going to go in without a little bit of effort. And so what will move without any effort is the water. See all this other stuff out here? This is all water, and they didn't actually draw it for you. So what you have to ask yourself whenever you learn these terms, hypo, iso, and hyper, is where is the most water? Where is the most of the solute? So the solute is whatever is actually dissolved. So like, for example, Kool-Aid. Kool-Aid, the powder mix, would actually be the solute in water. Okay? Food coloring could be the solute. Anything dissolved in water becomes the solute. And the thing that does the dissolving is called the solvent. And water's just the universal solvent. It's not the only thing that dissolves things, but it's the big dog. Okay, it dissolves more things than anything else. I can't really draw a dog. It always looks like a stick dog. Oh, that's horrible. Okay? When you're trying to decide what is the name of the solution around the cell, because this term right here is not referring to the inside at this point, it's referring to outside the cell. You have to look at the water level. So you have to say to yourself kind of the opposite. Well, I know there's a lot of salt here, and there's not very much salt here. So if there's not very much salt, there must be lots of water. So lots of water here, less salt. So anytime you have a lot of something, the prefix in biology for a lot of something, or above, not always a lot, but above, is hypo. And hypotonic solutions around the cell means that there's a lot more water on the outside than on the inside of the cell. Okay? Isotonic solutions, if you'll notice, we said before, there's equal amounts of salt inside and outside. Well, if there's equal amounts, or pretty much the same amount inside and outside, then there's probably about the same amount of water inside and outside. And so we would call the solution outside isotonic, because iso actually means same. Like you probably heard isosceles triangle, isomer, isotope from maybe 7th or 8th grade science. Those are terms that mean the same. So we have water the same amount on the inside and outside. Now over here, remember you said there was more salt on the outside than on the inside. So if there's more salt, then there's probably less water. I probably should have color coded this a little better for you. So if there's less water here, where is the more water? There's going to be more water inside. So because water is going to be the guy that usually moves, it's going to move outside. So whenever we have, and remember this term right here refers to the outside of the cell. So there's a lot of solutes out here, which is above what's inside. So remember, there's more salt than inside. So our word for above means hyper. Hyper means above. So if you think of people that are hyper, they usually are more active than the rest of us. We all wish we had that energy, right? So the kicker is you need to know what's going to happen to the cell as a result of this movement of water. So if this is the cell, and the cell is in a hypotonic solution, that means there's not very much stuff outside. There's a whole bunch of stuff inside. Where's there more water? So there's more water here, so the water will tend to go into the cell. Now when the water goes into the cell may have looked like this, but buddy, it's going to get really big as a result of that water going into it. It's going to swell up like a hippo. It's going to get big. Now in animal cells, they'll pop. Plant cells don't pop because plant cells have a cell wall around them. Now, the isotonic solution, we said, had the same amount of solute inside, or salt, or whatever's dissolved, as outside. And iso means equal. Now, I don't want you to get the misconception that nothing's going to happen here. Because what's going to happen is water is going to move equally in as it does out. So what do you think is going to happen to our lovely cell? Nothing. It should stay the same size whenever the water moves in and out at an equal rate. So that's a good thing. You want things to be isotonic to your body. Finally, hypertonic. Hypertonic refers to the fact that there are outside of the cell a whole lot more dissolved substances than inside. So this would be like I told you earlier, if you were out in the ocean and the salt water, your body's going, oh my gosh, look at all the water all the salt out there. Or if you drink a lot of caffeines or alcohol, then what will happen is your cells go, holy cow, we need a balance. We need to get the water to be the same inside as outside. And so because there's more water inside, the water's going to go out. So what's going to happen to your cell? It's going to shrivel up. And this is called dehydration. So the water's going to go out. Ms. Schindler likes to say that if you're hyper in her class, then you will go out in the hall. So I thought that was a great way to remember that. If you are hyper in class, you will get kicked out of the hall as a result. Now, you've probably heard of people dying from alcohol poisoning. Well, this is as a result of the water in their cells leaving. If you ever drank a lot of coffee and things like that, you have to go to the bathroom a lot. That's because your cells are trying to maintain a balance. And you can dehydrate. And people who drink too much alcohol in a short amount of time actually die from alcohol poisoning, which is basically dehydration. Alright, I've recorded this twice now, and it keeps giving me an error, so hopefully the third time is the charm. So, when you take your test, or do your short answer, or when you're in lab, you're not going to get to just look at lovely little pictures like this. Oh, you're not even able to see anything. You're just watching the screen. That's so lovely. You're going to have to look at, you're not going to be able to just look at lovely little pictures like this to predict what's going to happen to the cell. You're going to actually have to look at these types of diagrams where there's actually percentages involved. And so I need to show you how to do that so that you understand where the water's going and how to predict what's going to actually happen to cells when they're placed in various solutions. Now, there's 3 basic questions you're going to ask yourself. I'm going to kill the bird, by the way. She keeps sounding like a cat. I don't even have a cat. I don't know where she would have learned that. You hear her? So anyway, here's a cell placed in a beaker of water. And in this first beaker, we're going to put a 70% solute concentration. And the cell inside will be 20% solute. All right, I'm getting her some beans so that she can stop talking. Okay, inside of this cell, we're going to put a 40% solute and outside a 40% solution. Inside of this cell, we're going to put a 90% and a 10% solution outside. So remember, this is referring to the amount of solute, not the amount of water. None of these are referring to the amount of water. If we wanted to say water, we'll say water, but right now we're saying solute. Okay? So the first 3, in order to solve these problems and figure out where the water's going, there's 3 basic questions. The first one is, what is the solution, and I'm going to abbreviate this for the sake of time, outside the cell? So you have 3 choices. Is the solution outside the cell above concentration, equal concentration, or below concentration? If its concentration outside is above, what is our prefix for above? Hyper. And if it's equal, what is our prefix for equal? Iso. And what's it for below? Hypo. Okay? So the first thing you got to establish is, what is the solution outside? Second question, where does the water go? Where does it go as a result of this? Third question, what happens to the cell? Does it shrink? Does it shrivel? Does it stay the same? Okay, so let's start with our first one. All right, is the solution outside, above, below, or equal? It's above concentration. So we're going to call it what? A hypertonic solution. And like in hypertonic solutions like coffee, alcohol, the ocean, your cells will actually do what? They will lose water. Remember in Miss Schindler's class, hyper people go out in the hall. Your cells will lose water. So water loss, and the cell itself will actually shrivel up. Now in a plant cell, the cell membrane, because that's what we're talking about here, this purple thing we're drawing, will actually pull away from the cell wall. And when it does that, this is called plasmolysis, or the molded form being broken. And that's okay in plants for a little while. Like you might see a piece of celery that you've bought that's very limp, okay? Like you get it in the grocery store and it's kind of like not looking itself. Or maybe you buy a plant that hasn't been watered for a while. So this whole organism is said to be flaccid because it has very little water. So all you have to do is turn around and water it, and it will increase in turgor pressure and go back to normal. So that's in plant cells. Sorry, I diverged again. All right, let's look at the second one. Is the concentration outside, above, below, or equal? Okay, it's equal. And so when it's equal, what is our root word for equal? Isotonic. So we're going to call it isotonic. And the water's going to go in and out at the same rate. So it makes like a little equal sign. So same water rate. I'm sorry, these are fragments. And so your cell is going to pretty much stay the same size and the same shape. Last one. Is the concentration of solute above, below, or equal? Below. Very good. And what is our root word? Hypotonic. Hypo, hippo. Big, fat. So where's the water going to go? Into the cell. And the cell will start to swell. It looks kind of like a... Anyway. So the cell will swell. So water's going to go in. And the cell's going to get really big. Okay? If it's an animal cell, it will actually pop. This is called cytolysis because the cell is breaking. In plant cells, as you already know, they actually have that cell wall surrounding them. Oops. And so they're going to get bigger and bigger and bigger. Okay, which will be in the case where the plant gets more water, and when that happens, they get very turgid, which is a good thing. Okay, now what I want you to do really quickly is I want you to go back to your notes, and I want you to try to practice these 3 beakers with cells in them, and then I want you to decide if the solution outside is above, below, or equal. Then I want you to tell me, draw arrows where the water moves, and then predict what will happen to the cell, if it were an animal cell or a plant cell. So go ahead and pause this so that you're not just watching me do the answers, because that would be cheating. Okay. So the first one, is the solution outside above, below, or equal? It's below, so you should have said it's a hypotonic solution. Okay, where did you predict the water would go? Inside, because look, this is 100% water, so all that water has to go into the cell because it's trying to maintain that balance. This is kind of like that lady that tried to win a Wii for her child, and she drank so much water that the water went to her cells and they actually ruptured. So anyway, that's a hypotonic solution. The water's going to go in, the cell is going to swell. So in an animal cell, if it continues to swell, the cell swells in both cases, but in an animal cell, the cell will burst, and that's called cytolysis or cytolysis. Plant cells, they just become very turgid. You can kind of see a picture over here. All right, your second one. Is it the outside solution above, below, or equal? Equal. What's our word root? Isotonic. So in both situations, hopefully the cells will stay the same size. Okay, so this is like if you went to the hospital and you needed an IV. You want to make sure that IV was isotonic to your blood, or contact solution. Contact solution for your eyes, you want that to be isotonic. If you just use tap water, it'll burn your eyes and give you bacteria because it's not isotonic to your body. All right, your last one, is the outside above, below, or equal? It's above, so it's going to be called a hypertonic solution. And what happens to students when they're hyper? They leave the classroom and the cell will shrivel. So both cells will shrivel. In animal cells, they'll just get really, really small and the person will dehydrate. In plant cells, the cell membrane actually pulls away from the cell wall and the cells become very flaccid. And as an organism, if you actually went into a grocery store to look at the plant section, you would probably notice that if the cells are flaccid, your little plant would be kind of bent over like this, and you'd be like, oh, I don't want to buy that plant. It's all wimpy and limpy and all that kind of stuff. But all you'd really have to do, I know you should love my plant, is go back in and water it. And when you water it, the turgor pressure increases and the plant stands back up. Okay? So I know this was a long Vimeo, but I wanted to make sure you kind of got an idea over all of it, and hopefully it'll make more sense to you when you go to class and your teacher goes back through it a little bit more.